The role of DNA in any organism is to encode the genetic information. This happens in two ways. In normal life cycle, through transcription and translation, it may make proteins which catalyze most of the functions needed for daily life. It can be muscle contraction, brain activity, or other things. And as you've already seen, it's also replicated, copied during replication to pass on to the next generation.
Okay. Is there any problem with this process?
Well, not with the process, but it turns out that DNA is not an indefinitely stable molecule, but rather it can undergo changes with many chemicals.
So now, what are some of the chemicals that can damage DNA?
Many of them are parts of normal metabolism. For example, oxygen, which is essential for life. Unfortunately, it can form many reactive molecules, species that can damage the DNA. And as you know, our bodies are made up of water, 70% of it, right?
The water should be okay.
Well, turns out water can also react with DNA, albeit at a very slow rate. But given that we have in our bodies 37 trillion cells, and each cell has 3 billion base pairs, even slow reaction can eventually lead to changes in the DNA. DNA is also endangered by an environment. For example, sunlight catalyzes photoreactions that lead to DNA damage when you're at the beach. Some DNA damaging agents are made by humans.
What are they?
For example, when we barbecue meat, chemical reactions take place at the high heat that form chemicals that can react with the DNA. Industrial exhaust from cars or factories can damage the DNA. And the worst thing you can do to your DNA and health in general is to smoke cigarettes. Cigarettes contain dozens of chemicals that can react with the DNA. And indeed, we know that smoking causes lung cancer due to the damage influenced on the DNA by these compounds. Now let's look at one specific type of DNA damage, how it affects DNA and how cells have learned to counteract the damage so that it does not cause mutations. One of the most frequently occurring type of damage is 8-oxoguanine.
How does that happen?
8-oxoguanine happens by the reaction of oxygen with the 8 position of adenine, leading to a modification of the base. 8-oxo-G can pair normally with adenine, but it can undergo a bond rotation from the normal anti to the cis conformation.
So what is the problem here?
If you look at the cis conformation, the base pairing scheme just looks like it does for thymidine. So 8-oxoguanine in the syn conformation can form a base pair with adenine instead of cytosine, and when you do that, it can lead to a mutation. In other words, it can pair equally well with A as with C during replication and may induce changes like that.
Okay. So what happens in the cells?
In the cells, the following scheme happens. You get the oxidation of the G as we have just seen, and then as you undergo replication, the C part will do a normal GC base pair and go on, but the 8-oxo-G can now pair with an adenine. In the next round of replication, this adenine can pair with a T residue, leading to an AT mutation. So in the long run, the net result is that you go from a G to a T and have that change in the genome.
So mutation can alter the amino acid sequence of a protein.
That is correct. It may affect gene that regulates cell growth, for example, an oncogene or tumor suppressor, and that contributes to cancer formation. Fortunately, cells have evolved defense system to counteract that threat of DNA damage in the form of enzymes. So one of them for 8-oxoguanine is shown here. It's called 8-oxoguanine DNA glycosylase, or OGG1, and it can remove 8-oxo-G from DNA. In this figure, you can see the DNA in yellow and in orange with the damaged strand, with the 8-oxoguanine for emphasis shown in the sphere representation. The enzyme is green here, and the left showing the backbone trace, and here over on the right, just showing the surface of the protein in transparent green. Can you see how it flips out the modified base at the edge of the DNA into an active site of the protein? This binding pocket is specific for 8-oxo-G over guanine, and OGG1 will cleave out the 8-oxo-G so that it can be replaced again with a normal G residue and restore the original sequence.
It's repaired.
Now it's repaired. This is one of the many examples of repair systems that we have in our body to fix DNA, broken by various agents of natural and human-made origin.